This video is on question 44 of the 2011 IMAT paper. So in this question we're asked which of these following features are shared by both prokaryotic cells and eukaryotic animal cells, and we're given a list of glycogen, the cell wall, DNA end loops, and cytoplasm containing ribosomes. And on the right here I've got the potential answers. So before we start, let's ensure we actually know what we're talking about. So, prokaryotic, what does this mean? Prokaryotic cells are bacteria. They're the exact same thing. And eukaryotic cells are cells which have a nucleus. And so examples of eukaryotic cells are animal cells, plant cells, fungi cells. And there are some exceptions to of eukaryotic cells which don't have nuclei. Can you think of a well-known exception? The erythrocyte, or red blood cell. So, for answering this question, it can be useful to compare and contrast prokaryotic and eukaryotic cells for these features. So, let's start with glycogen. Can you tell me what exactly glycogen is? So, glycogen is a polysaccharide used for energy storage. And glycogen is found in prokaryotic cells, and it's used to store glucose. This glucose could then be used for glycolysis, and it's used for the exact same purpose in eukaryotic animal cells. Next, the cell wall is a structure found in multiple different cell types, and a few of its functions are for support and protection. So, is the cell wall found in prokaryotic cells, found in bacteria? Yes, and there's two types of cell wall, gram-positive and gram-negative, although that is likely to not be covered at A-level. Next, eukaryotic animal cells. Do animal cells have a cell wall? No. Animal cells just have their outer cell membrane, the lipid bilayer, which allows them to be more flexible than mature plant cells and prokaryotic cells. So our next feature is DNA in loops. Now, do you know a word which describes DNA in loops? Plasmids. Plasmids are literally just loops of DNA. And plasmids are found in prokaryotes, and they allow the transfer of DNA because the DNA in bacteria is stored in these rings of DNA, plasmids, and can be transferred between bacteria via an organelle called a pilus. Now, eukaryotic cells, they lack plasmids because the DNA in eukaryotes is instead stored as chromosomes, and a chromosome is essentially just DNA wrapped around these proteins called histones. Our final feature is ribosomes in the cytoplasm. Now, what are ribosomes? Ribosomes are an organelle found in all living things used for translation, that is, converting an mRNA strand into a polypeptide. Therefore, as they are found in all living things, ribosomes are found in both prokaryotic cells and eukaryotic animal cells. Therefore, we can see that feature 1, glycogen, is found in both prokaryotes and eukaryotic animal cells, and feature 4, ribosomes in cytoplasm, is found both in prokaryotic and eukaryotic animal cells. Therefore, the option which this satisfies is D, 1 plus 4. So our answer is D.